Welcome to part 23 of this video on the immune system. Okay, so we're currently in the process of discussing B-cell activation, which is a major part of the humoral adaptive immune response. Okay, so our B-cell here, directed against a certain epitope of an antigen of the pathogen, has now received both signal 1 and signal 2 for B-cell activation, and what's now going to start occurring is it's going to divide and divide and divide, and it's going to form what is known as a germinal center, and it's that I want to now show you. So quite simply, the B cell is going to divide and divide and divide and divide, and you're going to get a huge population of identical B cells that all have the same design for immunoglobulin molecules as the original B cell being produced. So you're getting clonal expansion occurring here. So that's a key term, so I'll just jot it down here. Now, the clonal expansion is very visible. If you actually look at the lymph node, you can see the deformation of the B-cell follicle where the germinal center is actually being set up, and that's what I want to show you in the picture in just a moment. So clonal expansion is going to occur, and we're going to produce a huge number of B cells that all have the identical design for immunoglobulin molecules, and therefore are all directed against the same epitope of this antigen. Okay, right, so now let me actually show you what a germinal center is going to look like. So I'm going to draw a picture of the lymph node that we will say is the lymph node in which our original B cell was present when it received signal 1 and signal 2 for activation. So I'll just draw a quick picture of this. So here is an afferent lymph vessel, here's another afferent lymph vessel, here's another afferent lymph vessel, and that can be the final afferent lymph vessel that I'll show for the basis of this picture, and here is the efferent lymph vessel. Then we have here the marginal sinus of the lymph node and the medullary sinus of the lymph node. And then this is the tissue where the lymphocytes are present. We'll have the cortex and the paracortex. So in the cortex will be the B-cell follicles. So here's a normal B-cell follicle, and now let me show you what a B-cell follicle will look like when it's had one of its B cells begin dividing and dividing and dividing, participating in this clonal expansion. So remember, when the B cell receives signal 2, it will be at the outer portion of the B cell follicle, because that's where it had to go in order to interact with the effector helper T cells. So now what you're going to get is this large sort of deviation of the B cell follicle, like so, on the edge, which is where you've now got a huge, great new population of B cells being produced there, all from this single B cell that receives signal 2 of B cell activation. Here's another normal B cell follicle, and here's another normal B cell follicle. So let me just colour this in. So I'll colour the normal B cell follicle tissue in green here. So these are all of the different B cell clones that are currently in the inactive state and haven't received signal 1 and signal 2. And then here is the germinal center on the side of this B cell follicle here, and that's made up of all of the progeny of this one B cell that was activated by the effector helper T cells and received signal 2, and therefore started to divide and expand its B cell clone. Okay, so this is a population of all identical B cells that have the same design for the immunoglobulin molecules, and therefore are all directed against the epitope of the antigen. Okay, right. So that's what is meant by a germinal center. What I now want to discuss is some processes that are going to occur in the germinal center. I want to discuss the process of somatic hypermutation and the process of class switching. And then finally we'll come on to how the germinal center is actually going to produce effector B cells, which are also known as plasma cells, and of course memory B cells as well. And then we'll discuss what the effector B cells or plasma cells are then going to do to actually contribute to the eradication of the pathogen. Okay, so firstly then, I want to describe this process of somatic hypermutation, and this is another quite complicated topic, so you're probably absolutely fed up of complicated topics in this video on the immune system, but I'm afraid there is this next complicated topic, and class switching is another quite complicated topic, so we have still got some complicated topics to cover. So somatic hypermutation is the next one that I want to go over. So what is actually going to occur is that in these different B cells that we're producing, so we produce this germinal center, which is all the progeny of this original B cell that was directed against that epitope of the antigen that we're launching this humoral adaptive immune response against. What's now going to occur is in these progeny here, you're going to get slight mutations occurring in the actual B cell receptor genes. So in the immunoglobulin light chain and also in the immunoglobulin heavy chain. So what's going to actually start occurring is these different B cells in the germinal center are all going to start changing their immunoglobulin molecules so that they all start getting small variations in their immunoglobulin molecules. So remember, the immunoglobulin molecule is made out of heavy chains and light chains, and each of those two chains has a variable region and a constant region. I'll just draw a quick picture of this. So here's our basic picture of an immunoglobulin molecule, and remember, if I just draw these dashed lines here, these are the variable regions. So this is the variable region of the immunoglobulin light chain, the variable region of the immunoglobulin heavy chain, and it's exactly the same over here. This is the portion that actually makes the antigen binding site. And what's actually going to occur is that the progeny of this original B cell are going to start mutating their genes for the immunoglobulin heavy chain and the immunoglobulin light chain. And when I say the genes, I mean the immunoglobulin heavy chain locus and the immunoglobulin light chain locus that they are actually using to make the immunoglobulin heavy chain and the immunoglobulin light chain. So the loci which successfully underwent VDJ recombination, they are going to start introducing point mutations into the portions of those loci that code for the variable region of the immunoglobulin heavy chain and the immunoglobulin light chain. And these mutations will be point mutations. Okay, so what this will lead to is actually all of the different cells in the germinal center will now start to actually have slightly different immunoglobulin molecules. They'll all be based on the original immunoglobulin molecule of the original B cell, but they're now all going to start having different, slightly different immunoglobulin molecules. So now the question is, well, what on earth is the point of this? So the answer as to what the point of this is that means that the germinal center is now going to have a range of different B cells that have slightly different immunoglobulin molecules, slightly different antigen binding sites on those immunoglobulin molecules, but they're all roughly based on the original immunoglobulin molecule that had an antigen binding site that could recognize the epitope of the antigen. And the idea is now that some of these B cells in the germinal center, and I just like a little picture of the germinal center, so let me just draw a huge number of cells here so that I've got something to point at. So this can represent a portion of this germinal center here. So these are representing the cells of the germinal center, and I'll just color them in red here. And of course they would have nuclei, but I'll just draw their cell membranes. It will suffice for the purpose of this picture. Okay, so this is a little picture of part of this germinal center here. So what's now going to happen is all of these different B cells that are in the germinal center now, they're going to have slightly different immunoglobulin molecules now, because occasionally, and I do mean this very occasionally, point mutations are being made in these B cells in the germinal center in the portion of the immunoglobulin heavy chain locus, which codes for the variable region of the immunoglobulin heavy chain, and the portion of the immunoglobulin light chain locus, which codes for the variable region of the immunoglobulin light chain. And that's changing the antigen binding region of the immunoglobulin molecule that B cell can produce. Okay, so you now have variation in this population, and some of them will now produce immunoglobulin molecules that can bind more strongly to the specific epitope of the antigen that the original B cell was against. So some of them are actually going to improve their antigen binding site and make it better. Of course, some of them will make it worse. Some of them will put point mutations that will change the variable region of the immunoglobulin molecule for the worse, and it will now not be able to bind as strongly to that epitope of the antigen. So the idea now is that we are going to try and improve the affinity of the immunoglobulin molecules for the antigen by selecting for the B cells in this population that have got the higher affinity immunoglobulin molecules. So the idea now is that we are going to put a natural selection process in place so that the ones which have the best immunoglobulin molecules now, the highest affinity immunoglobulin molecules, i.e. the ones that have put in point mutations into their immunoglobulin heavy chain and immunoglobulin light chain that have improved the affinity of the antigen binding region for that epitope of the antigen. We want them to survive, and the ones that have made it worse or which have not made it any better, they're going to die. So we want a selection process to start up in the germinal center that's going to lead to our population of B cells gradually getting higher and higher affinity immunoglobulin molecules for that epitope of the antigen that we are launching this humoral adaptive immune response against. Okay, so let me now describe how this selection process is going to work, and it's a very simple idea here. So the selection process, the way it's going to work is that in order for these B cells to continue surviving, they need to continually receive more help from the T cells, from the effector helper T cells. So in order to get help from effector helper T cells, what do you have to do? You have to put on your surface fragments of the antigen that the effector helper T cells can recognize on MHC class II molecules. Then you can form one of these immune synapses with an effector helper T cell, and then the CD40 ligand from the effector helper T cell can bind to your CD40 receptor and give you a signal to survive and divide. In addition, the effector helper T cell might be a T helper 2 or a T follicular helper effector helper T cell, and it will then give you lots of interleukin 4 molecules to further stimulate you. However, if you don't get T cell help, then you won't get this sort of stimulation, you won't get the activation of the CD40 molecules, and you will end up dying. So that's how this selection process is going to work. To survive, you have to get T cell help. You have to therefore be able to present fragments of the antigen that you're supposed to be directed against on your surface. So how are you going to get those fragments? Well, of course, to get those fragments, your B cell receptors on your surface, the immunoglobulin molecules on your cell surface, have to be able to bind to antigen molecules on pathogen fragments that are arriving in the lymph node, and then you can phagocytose them, break them down, process those antigens and put the fragments on your surface. So hopefully it might all start to be making sense in your head. We are making this massive great population of B cells, and they're all now getting slight variations in their immunoglobulin molecules. They're all getting slight variations in their B cell receptor. Now, they're all in competition for antigen. The antigen molecules are coming to the lymph node from the infected site, i.e. it's being drained by lymphatic vessels, and the lymphatic vessels are bringing antigen molecules on fragments of the pathogen to the lymph node. But this is scarce. There's not enough for all of the B cells in the germinal center. So which B cells in the germinal center are going to get those antigen molecules on those pathogen fragments? It's going to be the ones that have improved their immunoglobulin molecules antigen binding site for the better. It's going to be those ones which, when they participated in the somatic hypermutation process, improved the immunoglobulin antigen binding site and made it have a higher affinity for that epitope of the antigen. They are the ones which therefore will actually get the antigen molecules. They'll phagocytose those antigen molecules attached to the pathogen fragments, and they'll process it and present it on MHC class II proteins, present the fragments on MHC class II proteins. They'll then migrate to the edge of the germinal center and interact with helper T cells and get signals to survive. So the ones with the highest affinity immunoglobulin molecules, or B cell receptors, they will get the signals to survive and divide, and they'll produce loads of progeny which will all have this improved version of the immunoglobulin receptor. Sorry, the immunoglobulin molecule. I keep saying receptor. They'll all have this improved version of the immunoglobulin. Okay? And then the process will continue. It won't stop there. Their progeny will continue making somatic hypermutations and either improve the immunoglobulin molecule further or go back a step. Meanwhile, those cells in here that do not have good immunoglobulin molecules, i.e. they've suffered somatic mutations that are making the immunoglobulin molecule worse at binding to that specific epitope of the antigen molecule, they will not do this process. They will not get antigen, they will be outcompeted, and they therefore will not present fragments of the antigen on their surface to helper T cells. They will not get that signaling, and they will die. So hopefully you can understand how over time, if that process goes round and round and round, and it does continue going round and round and round, this is what's happening in these germinal centres. This goes on and on and on, cycle after cycle of B cells getting antigen from these pathogen fragments, presenting the fragments of the antigens to helper T cells, and then getting signals to survive and reproduce, producing a whole population of B cells which have a similar immunoglobulin molecule to them, but then their progeny are all going to undergo the somatic hypermutation process as well, so you'll get further variation. And hopefully what you can appreciate is, as this process goes on and on and on, overall, if we were to look at the germinal centre, what's going to happen is, as a population of B cells, overall the immunoglobulin molecules in that population, although there's going to be variation, overall the quality of the immunoglobulin molecules is going to go up and up and up, okay, because of this selection process. We are selecting for B cells which have immunoglobulin molecules with a high affinity antigen binding site for this epitope of the antigen. Therefore, those ones will survive and reproduce, and the ones which don't will die. It's a natural selection process. It's a classical Darwinian natural selection. And therefore, over time, the population will evolve, it will change, and the characteristic that we're looking at is the affinity of the immunoglobulin molecules for that epitope of the antigen, and gradually the population will evolve to have a higher affinity immunoglobulin molecule overall. And this process is known as affinity maturation. So the affinity of the immunoglobulin molecule is going to improve for the epitope of the antigen. So somatic hypermutation in germinal centres leads to affinity maturation. So this is one of the key things that's occurring in B cell germinal centres. And we will come on to the actual production of effector B cells. At the moment, these B cells inside a germinal centre are not effector B cells at the moment. They're still mature B cells, but they're not yet effector B cells. We'll come on to the differentiation process into effector B cells. At the moment, we're just trying to make it so that when we do produce effector B cells, we're producing them with as high affinity immunoglobulin molecules for the epitope of the antigen as is possible. Okay, right, the next thing that I want to discuss is that class switching can actually occur inside B cell germinal centres. So the B cells that are inside a B cell germinal centre can actually change the class of immunoglobulin molecules that they are actually producing. They can change which constant region they are actually going to use for their immunoglobulin heavy chains. So remember, initially the mature naive B cell, it just produced immunoglobulin heavy chains of the IgM and IgD membrane bound type. What's now going to happen is the B cells in this germinal centre, some of them will start actually changing that, and they'll decide, actually we don't want to produce IgD and IgM type immunoglobulin heavy chains, we want to produce different types, and this is known as the process of class switching. So, alongside the point mutations that are occurring as part of somatic hypermutation, they can start to actually recombine their DNA, they can cut huge bits out. So this is a very small modification where you're just changing one organic base for another organic base. Class switching is going to involve something much similar to VDJ recombination, where you're going to cut out a huge, great portion of the DNA. I should have actually stressed more what I meant by point mutation. A point mutation in the context of somatic hypermutation up here, that means that you change one organic base for another organic base. So you substitute one organic base for another organic base. That's what we mean by point mutation. So it's a very small change to the DNA. You're making a very slight change to your immunoglobulin heavy chain or immunoglobulin light chain variable region. Okay, now in class switching you're going to make a much bigger change, and to understand this we need to bring back up this picture of the immunoglobulin heavy chain locus. So after VDJ recombination, this is what the immunoglobulin heavy chain locus is going to look like. So this is what the immunoglobulin heavy chain locus will look like in all of the B cells in our B cell germinal center. Just imagine having all of this portion from this orange constant gamma 3 gene segment all the way up to this green constant alpha 2 gene segment inserted in there. Okay, and this is the region that they'll be introducing point mutations into, where we've got the selected variable diversity and joining gene segment, which codes for the variable region of the immunoglobulin heavy chain. Now, what can happen in these B cells in the B cell germinal center is that they can start cutting huge bits of the DNA out, and this is how they're actually going to change which immunoglobulin heavy chain constant region they're actually going to use. So you can't just start transcribing for a greater distance. That's not how we start making the different immunoglobulin heavy chain constant regions. So you might think that all we need to do is just transcribe for longer so that the pre-mRNA has all of the different constant gene segments in, and then we can do the change as a splicing change, and that's how we can start expressing the other forms of immunoglobulin heavy chain. That's not the way it actually works. What actually happens is they start cutting a huge amount of their immunoglobulin heavy chain loci out. So let's just do an example. So let's say we are going to have a B cell which is going to decide that it would like to make heavy chains of the immunoglobulin G2 form. So let's say we've got a B cell in the germinal center that wants to start producing heavy chains of the IgG2 form. What would happen to do that is it would cut all of the DNA from the constant mu gene segment all the way up to the one in front of the constant gamma 2 gene segment, so all the way up to here. So it would cut out the constant alpha 1 gene segment here. It would cut all of this out, so all of this would now be removed. So it would cut that bit out, cut here and somewhere here, like so, and it would cut all of this bit out. That bit would be binned, and then it would bring this constant gamma 2 gene segment right up to here. And now in the transcription process, and I should probably be showing this on this picture here, remember that the portion upstream of the constant gene segments would look like this, not this. Okay, now in the transcription process what will occur is you'll still transcribe the variable diversity and joining gene segment portion here. You'll transcribe all of these joining gene segments as before, but then your constant gene segment would now be the constant gamma 2 gene segment that you'd have in the pre-mRNA, and then you would stop transcription after the constant gamma 2 gene segment. So I know for constant mu you went past that and then also got constant delta. That's the only one where you would go past it. For all of the others, so if you brought any of these other 9 constant gene segments into position 1 here, transcription would stop after that one had been transcribed. It's only for constant mu that you go past it and then also end up transcribing constant delta, and then at the end of constant delta you end transcription. For all of the others you end transcription after they've been transcribed. So if you want to produce any of the others you have to bring them into position 1. So in this case you bring constant gamma 2 into position 1 and it would then get transcribed here. Transcription would end there, and then you'd splice out all of this intermediate bit and you'd end up with a mature mRNA where you'd have the variable diversity joining gene segment, and then you'd have the constant gamma 2 gene segment piece of RNA here, and that would now be translated to form you an immunoglobulin heavy chain where the constant region was the IgG2 type. Okay, so this is what can also happen in these B cells that are in the germinal center. They can start cutting bits of their immunoglobulin heavy chain locus out, and that will mean that they'll start producing immunoglobulin molecules of these different types. Okay, and that process is known as class switching. And I want to stress that, remember, each of these constant gene segments at its terminal portion will have a region that codes for the membrane implanting portion of the immunoglobulin heavy chain. And that portion can either be left in at the splicing process or it can be spliced out. And initially these B cells in the germinal center are all going to leave that in. They're not going to produce secreted immunoglobulin molecules until they are actually effector B cells. So only effector B cells produce the secreted immunoglobulin molecule, and therefore only effector B cells cut out the final portion that inserts the immunoglobulin heavy chain into a plasma membrane. So they're going to produce membrane-bound IgG2 in this case. So if the B cell if one of the B cells in the germinal center was to do this, it would now produce on its surface IgG2 membrane-bound immunoglobulin molecules, and they would now form the role of the B cell receptor, and they can do the job of the B cell receptor just as well as the IgM and IgD molecules that we had previously could do it. Okay, so all the different classes of immunoglobulin molecules, they can all perform the role of a B cell receptor. It's just that initially in the naive B cells, before we've had germinal center formation, it's just the IgM and IgD forms that are actually produced. But now in the germinal center, after we've had class switching occur, they can have B cell receptors of the different types actually present. We will see that as secreted forms, the different classes of immunoglobulin molecules do slightly different functions, but as B cell receptors, they're all equally good at performing that role. Okay, so those are the two processes that are occurring inside germinal centers in lymph nodes. I'm going to end this video here and we'll discuss the final portion of this great humoral adaptive immune response in the next video, which is the differentiation into effector and memory B cells, and then what the effector B cells are actually going to do. But just before we do, just before we have a break, let me just summarize what we have seen about germinal centers. So, once your naive mature B cell receives signal 2 from the effector helper T cell, it is going to divide and divide and divide, i.e. it's going to clonally expand. It's going to expand that B cell clone. So you're going to end up with loads of B cells that all have immunoglobulin molecules very similar to the original B cell. Now, why do I say very similar rather than identical? Well, because of the process of somatic hypermutation. We form a germinal center from all of these new B cells that are the progeny of this original B cell, and this sticks out on the side of a B cell follicle like so. So they're very obvious when you look at a lymph node histologically. And what's going to occur inside germinal centers is a process called somatic hypermutation. And the reason it's called somatic hypermutation is that it's mutations occurring somatically. Okay, and they're occurring very fast. They're not occurring at the normal rate that mutations would occur. So of course mutations do occur all the time in your DNA. It's unavoidable. Mutations do occur, and there is lots of machinery inside cells to repair the DNA after they do occur. Somatic hypermutation is a very fast process of mutation which is caused deliberately. It's done deliberately within the cells. So you get mutations introduced to the immunoglobulin heavy chain and the immunoglobulin light chain genes, specifically into those portions that code for the variable region of the immunoglobulin heavy chain and the immunoglobulin light chain. These are very mild mutations that are only going to result in very mild changes to the immunoglobulin molecules that you're producing. They are point mutations where you just substitute one organic base for another organic base, and you'll know that the biggest thing that can lead to the biggest thing that could potentially lead to is a premature stop codon. In that case, the B cell that you get is just going to become totally useless, so it will die out in the selection process. But normally what that's going to lead to is just one amino acid in the immunoglobulin heavy chain or immunoglobulin light chain being swapped for another one. Okay, and that will change the immunoglobulin molecule only very slightly. So now what we're going to get in our germinal centre then is a huge amount of variation in the population of B cells. So we'll have loads of B cells now, and they've all got slightly different immunoglobulin molecules, all based on the immunoglobulin molecule of the original B cell, which had an antigen binding site that could recognise this epitope of the antigen that we're launching an adaptive immune response against, but all slightly different. Some of them will have now a lower affinity for binding to that epitope of the antigen. Some of them will have an improved affinity for binding to that epitope of the antigen. We are now going to set up a selection process to select for those that have an improved affinity for the epitope of the antigen. And this is quite simple. Those which combined best to antigen molecules will survive, and those that don't will die. Okay, and the actual technicalities of how this works is that we've got these fragments of pathogens or entire pathogens coming up to our lymph node, but they're in scarce supply, and of course those B cells that have the highest affinity immunoglobulin molecule, that they are most likely to actually bind to those antigens on the fragment of the pathogen or the entire pathogen, and they will then phagocytose the fragment of the pathogen or the entire pathogen. They'll process the antigen, and they'll put the fragments on MHC class II molecules. They'll then migrate to the outside of the germinal centre and receive T cell help. They'll receive the activation by the effector helper T cells. They'll form one of these immune synapses, and they'll get CD40 activated by the CD40 ligand on the effector helper T cell. They might also get some interleukin-4 if they're lucky. That will give them the signal to survive and also reproduce. Okay, so they'll produce loads of progeny that are all based on them. And again, their progeny will now also be participating in the process of somatic hypermutation. Overall, you can hopefully appreciate that if this process goes on for a long period of time, then gradually the population is going to evolve. You are selecting for those B cells that have the highest affinity immunoglobulin molecules. So the population will evolve, and this is a process known as affinity maturation. So gradually the overall population are going to get more and more higher and higher affinity immunoglobulin molecules for that epitope of the antigen. Okay, so affinity maturation is occurring inside the germinal centre. At the same time, some of the B cells will start class switching, so they'll stop using IgM and IgD as their B cell receptor and start using other classes of immunoglobulin molecule as their B cell receptor. And that's a non-reversible change. You can't change that afterwards because you have chopped out a bit of DNA. You can change it further, and let me just explain this. So once the molecule once the B cell has decided, for instance, that it's going to make IgG2, it can actually decide that it would like to make IgE1, for instance, because it can chop further DNA out. It could say, okay, let's chop this bit out now. Once it's brought this to position 1, it could say, now we'll chop this bit out and we'll bring the constant epsilon 1 to position 1. So you can change it further, but of course you can't change it to one of these ones that's been chopped out. Once the DNA has been chopped out, it's gone. You can't ever use that DNA again, but you can perform further choppings to go to one that's downstream of the one that you've chosen previously. So if I was using IgG2, I could move to using IgE1 or indeed IgG1 or IgA2, but I couldn't switch to using, for instance, IgG1 here. Oh, that's confusing. Oh, that's IgG4. Sorry, I was misreading that. I thought we had two IgG1s for a moment, but no. So I could move to using IgG4. I have to apologise. Okay, so I hope that's clear. So class switching will occur, and we'll start getting B cells that are using different classes of immunoglobulin molecules for their B cell receptors. And as I say, you can change that still, but you can't ever get the DNA that you've cut out back. You can just cut more out and therefore switch to using constant gene segments that are further down from the constant gene segment that you previously used. Okay, so we will call it there for this video, and in the next video we'll talk about the differentiation process that's going to follow from the germinal centre.